Hello everyone, and welcome to Lecture 2 of our Microbial Genetics chapter, which is Chapter 8 in our textbook. In this lecture, we will be talking about DNA replication and binary fission. So in the first lecture of this chapter, we talked about one of the functions of DNA, which is to code for proteins. And in this lecture, we will be talking about the second function of DNA, which is to pass information off to subsequent generations. Here you have your lesson objectives as always. By now you guys probably know how to use those to your advantage. There is one I want to specifically point out, and that is being able to differentiate between DNA replication and gene expression. In the last lecture we talked about gene expression. In this lecture we will be talking about DNA replication, and you want to make sure you are clear on the steps of each, what each process is used for, and that you don't mix them up. I point that out because over the years I've had many people mix up the process of gene expression with DNA replication. And although there is some vocabulary overlap, they are two totally different processes used for totally different things in living organisms. So please pay special attention to this objective and make sure you are 100% clear on the difference between DNA replication and gene expression. For a prokaryote to be able to reproduce itself, it first has to replicate its genetic material. So that means, at a minimum, it must make a copy of its chromosome. If it has plasmids, it may copy those as well so that they can be passed on to the new generation. But sometimes things go wrong with plasmids and they don't always get replicated correctly, or they don't get passed off to the new generation. But at a minimum, the chromosome must be replicated so that when cell division occurs, the original cell keeps a copy of the chromosome and the new cell also has a copy of the chromosome. So this aspect of DNA really is talking about the second aspect of that molecule, which is heredity. And so in this lecture, we're talking about how DNA is passed from one generation to another, or basically how information about what proteins to create is passed from one generation to another. The goal of DNA replication is to create a second copy of the chromosome to pass off to offspring. At the beginning of the process, there will be one chromosome and potentially a couple of plasmids. And at the end of the process, there will be two chromosomes, so two copies of the same chromosome, and maybe copies of the plasmids as well, but maybe not. The key thing really is making sure that chromosome gets replicated. Replicating DNA happens in three main steps. The first step is for the DNA that is what we call in a supercoiled state to get relaxed and straightened out. So if you were to look at a picture of all of the DNA in, let's just say, an E. coli bacterium, and you were to straighten all of that out, it would be bigger than the bacterium actually is. So for DNA to actually exist inside cells, it has to be wound up really, really tightly and put into a really, really compact state. And this state is called being supercoiled. So the DNA is supercoiled most of the time for prokaryotes. And to be able to actually be replicated, that supercoiling has to be kind of undone and spread out. If you were to look at a ball of yarn or a ball of string and it was all wound up, like when you first buy it and it hasn't been taken apart or anything yet, that would be analogous to what the supercoiled DNA is like. Now, if you want to make something with that yarn or that string, you would have to undo it from that ball to be able to actually use it. And that's the same with the DNA. To be able to copy the DNA, it has to be taken out of that really, really tight ball that it's wound up in. There are a couple of enzymes involved in this first process of relaxing the supercoiling and straightening out the helix, and that is DNA gyrase, also called topoisomerase 2, and helicase. DNA gyrase does most of the work with relaxing the supercoiling and kind of taking it out of that ball form and straightening it out and kind of stabilizing the molecule during that process. And then helicase is going to go ahead and separate the backbones from each other. Once a chromosome has had its supercoiling relaxed, it's kind of straightened out, the helicase has separated out the backbones, this second stage can occur. During the second stage, the code on each strand of DNA is copied, essentially. The process starts with a molecule called RNA primase, and this molecule, this enzyme, is used to create a short strand of RNA that will attach to the DNA that DNA polymerase can use to grab onto the DNA. And that sounds a little bit confusing, but we'll go through it step by step in just a second. So primase is an enzyme that creates a short strand of RNA, and that short strand of RNA is called a primer. And that primer is going to attach to the DNA molecule. So I'm just going to put it in green here. There's going to be one on each strand of our DNA. So these are primers. And they are, like I said, short strands of RNA that the DNA polymerase will grab onto so that it can move forward and start replicating the code. In prokaryotes, since they have circular chromosomes, there is one molecule of DNA primase going clockwise. So I'm going to draw that here. So we've got one molecule of DNA primase going clockwise, and one molecule of DNA primase going counterclockwise. And eventually, they're each going to get their job done and meet in the middle on the opposite side of where they started. So if they start at what's called the origin of replication, each of them will copy about half of the DNA molecule, and they'll meet at this termination point. Once the DNA strands have been copied completely and DNA polymerases meet in the middle at this point of termination, then step 3, the final stage, can begin. In this stage, everything is pretty much done. There are a few final things to finish before we can say that the new DNA chromosome is complete and everything's ready to go. And there are 3 main enzymes involved in that process. There's DNA polymerase 1, DNA ligase, and topoisomerase 4. DNA polymerase 1's job is primarily to check these new DNA chromosomes, these new DNA molecules, make sure there aren't any big mistakes in them, maybe fill in any gaps that were left over from the primers. So when DNA polymerase 3 grabs on to the DNA molecule, it can't actually reach the nucleotides that are covered by the RNA primer. So those RNA primers have to be removed, and then those base pairs, those nucleotides, have to be filled in by the DNA polymerase 1. So that's kind of the job of DNA polymerase 1. Check for mistakes, fix any mistakes potentially, fill in the gaps from the RNA primers, and just check and make sure everything is basically filled in. There aren't any big spaces. DNA ligase will go and kind of seal up any places that the DNA polymerase 1 has had to fix. So if the DNA polymerase 1 had to add in nucleotides, DNA ligase will go and check and make sure everything is sealed, those nucleotides are in the right place, and they're not going to go floating off into the cytoplasm somewhere. And then topoisomerase 4 will actually separate out the 2 new complete chromosomes from each other. So it wouldn't do the cell any good if it got everything copied and everything was right. However, the chromosomes couldn't be separated from each other. So topoisomerase 4's job is to separate out the 2 new chromosomes from each other so that they can be given to new cells during the process of cell division. In prokaryotes, this process of cell division is called binary fission, and most of the time, prokaryotic reproduction leads to exact copies being made. So typically the parent cell starts out, replicates its chromosome, and then the cell is divided, and the 2 cells that arise from this one cell are exact copies of each other, and those copies are called clones. When you get enough clones together in one particular area, especially on a petri dish, that's often called a colony. So by now you guys have worked with lots of colonies in lab, and that is how those colonies arose. When you inoculated a plate for the first time, you couldn't see any bacteria on it. But over time, maybe over 24 hours or 48 hours, the bacteria that were on that plate went through this process of DNA replication and binary fission. They made copies of themselves called clones. And then once there were enough clones all together, you could start to see them, and you could start to see what's called a colony. So in terms of the actual process of binary fission, it's fairly straightforward, especially when you compare it to mitosis in eukaryotes. In step 1, the origin of replication for the chromosome is located. Step 2, the chromosome is copied. So all of those details we just went over on the previous slide, that's what happens here in this step 2 here of binary fission. Step 3, the new chromosome is finalized and the chromosomes are separated from each other. Again, we went over the details on the previous slide. Step 4 is when the cell actually has to cut itself in half. And it does that most of the time by creating what's called a Fit-Z ring that pinches in the center of the cell and splits it into two. So Fit-Z, as you remember, is one of the cytoskeletal proteins. And when a bacterium is going through this cell division process, it's going through binary fission, that Fit-Z ring is created. Basically, a bunch of proteins are put along the center of the cell, and those proteins contract just one step at a time. And every time they contract, the ring gets a little bit smaller. And as it gets smaller, the cells start to pinch in the centers, and eventually they split into two. So step 5, the clones separate from each other, and you have 2 cells from 1. I do want to do a quick review of mitosis because remember, we do study some eukaryotic microbes in this class. Most of our focus is on the prokaryotes, and then to a second extent, especially in the disease unit, the acellular microbes. However, we do have some eukaryotes that we focus on, and so I just want to do a quick review of mitosis so you can compare and contrast how prokaryotes reproduce versus how eukaryotes reproduce. Eukaryotic cell reproduction is called mitosis, and most of the time, in this case as well, they also produce exact copies of each other. So the parent cell typically replicates all of its DNA and then creates an identical daughter cell. In terms of the steps of mitosis, there are multiple phases. If you compare binary fission to mitosis, binary fission is much more straightforward, it's a lot faster, and more cells can be made in a shorter period of time than mitosis can do. So remember, with mitosis, you have to have multiple phases. Part of this is due to the fact that there is a nucleus and that there are multiple chromosomes. So prophase occurs, prometaphase occurs, metaphase, anaphase, telophase. And during all of this process, all of these different steps, what is happening is the DNA is replicated, the chromosomes line up, they get pulled apart, they get separated, and then eventually 2 new cells will form through the process of cytokinesis, which is when the cells actually separate from each other. One of our big objectives of the course is just to make sure that you can compare and contrast prokaryotes to eukaryotes. So we've done that before in cell structure, we've done that in terms of transcription and translation, we'll do it here with binary fission and mitosis, and then a couple of other times in the class as well, because it really is important to understand the difference between prokaryotes and eukaryotes. You need to understand how they're the same, but also how they're different, because the differences between prokaryotes and eukaryotes really are the things that allow us to treat bacterial infections effectively. So, coming back, bringing that all back to binary fission and mitosis. In terms of similarities, both begin with 1 cell and end with 2. The DNA has to be replicated first in both cases. If the DNA is not replicated or is not replicated properly, then you would end up with a cell division event that does not pass off all of the information, or at least the necessary information, to the next generation of cells. And in both cases, at least hypothetically, identical copies of the cells are made. So whether you're talking about clones, in the case of bacteria, you can see sometimes they are called daughter cells as well, but most of the time they're called clones. Or you're truly talking about the daughter cells that are formed during mitosis. In both cases, hypothetically, we have one cell that splits into two and creates two identical offspring. For differences, binary fission is typically a lot faster. This is not always true. There are some very slow replicating bacteria out there, but for the most part, binary fission is much, much faster than mitosis. Additionally, binary fission always reproduces an entire organism. So remember, prokaryotes are single-celled organisms, which means every time they go through binary fission, they are making a copy of a cell, but since that entire organism is just one cell, this is how prokaryotes reproduce themselves too. Binary fission is not just the process of reproducing a cell, it is also the process of reproducing an entire organism. And in some cases, when you're talking about unicellular eukaryotes, mitosis may also reproduce an entire organism, but in many cases, eukaryotes are multicellular, and so mitosis may not reproduce the entire organism, it may just reproduce one cell within that organism.